Muga silk, which is Muga is the costliest silk present here, and it is cultivated in. It is important for the rural economy of Assam. This is the Som plant. Som is Persea bombicina, and it is widely cultivated in, especially in Upper Assam and some parts of Lower Assam. And there are many plants which can act as host for the insect, which gives golden muga silk. But Som is the plant which is considered as the primary host plant because the nutritional quality of the leaves, it is very, very important for the growth and development of the larvae, growth and development of the insect, and consequently the quality of the silk. So this Som, which is Persea bombicina, this is capable of supplying all the nutritional requirement to the insect so that the silk is grown here. This is this plant, it gives good quality cocoon, and therefore quantity of raw silk material is also very high. These are the insects. Now, since it is a perennial plant, so it is exposed to different pathogens, and these pathogens, they especially attack the leaves. Since leaf is the food material for the insect, so there is significant loss in the leaf yield. I am not showing the data, but we had done one part of the work where we had calculated the incidence of disease and loss of leaf in different seasons of the year, and we what we found was there was a loss of yield to the extent of 20%. And since the leaves are infected with pathogen, so what happens is the quality of leaf is degraded. Since the quality of leaf is degraded, the insects, they do not get adequate nutrients for their growth and development, and consequently the quality of silk and the quantity of raw silk is reduced. So these are the three important diseases. First disease is grape blight disease, which is caused by Pestalotiopsis disseminata, and you see this is responsible for the highest loss. The loss varies from 13 to 42%. Then Phyllostricta persei, the loss is approximately 9%, and then Coletotrichum geosporidis, the loss is to the extent of 6.3%. So this data, it clearly indicates that the incidence of these three diseases, they really reduce not only the quantity of the leaf, but also the quality of the leaf. So these are the pure culture of these organisms. Now, traditionally, if you go through the areas where this Muga silk host plant is, or Muga silk is practiced, then you will find that traditionally the farmers, they are using Bavestine, which is the fungicide. But the problem is there are reported problems where it has been observed that this Bavestine, if it is not used properly, then it is also damaging. It is also is responsible for the harmful effect on the silkworm. And since it affects the silkworm, so it cannot be ignored. And then what is the way out? The way out is biological control. And in the last few years, a lot of work has been done in the area of manipulation of rhizosphere. So we thought that why can't we develop a microbial consortium? Our objective was to have a plant growth promoting rhizobacteria, a plant growth promoting fungi, and mycorrhizal fungi, because I am basically working on arbuscular mycorrhizal fungi. So I thought of incorporating all these three so that the problem of foliage disease of silk can be tackled. Now, our another idea was to induce resistance in the host plant by incorporating these three groups of organisms, that is PGPR, then PGPF, and mycorrhizal fungi. So I will not go into, it is not required, how the systemic resistance is induced in the host plant. But that was one of the idea which we had conceptualized, and so no point of going into the detail of this. So what we did, before we put these organisms into the pot, first we did the pot experiment. So before we put them into the pot, we had gone for, we studied the rhizosphere competence of microbes, like whether they are capable of, especially in case of bacteria, whether they are capable of forming biofilms or not, bacteria, whether they are capable of motility or not, whether they can really colonize the roots or not. And then if we are, because I told you we will be putting bacteria and fungi together, so whether they are biocompatible or not. So these three conditions means all these characters were evaluated before the bioformulation was made. So these are the three organisms. After a lot of isolation and screening, we shortlisted Pseudomonas, then Trichoderma harzianum, and Glomus. There was one Glomus species which could not be identified. All these three organisms, they have been isolated from the field, not from, they have not been taken from anywhere else. All these are indigenous organisms isolated from Soam plantation soil. So we have multiplied them. This is Trichoderma harzianum. The first one was Pseudomonas, and this is Globus species, which we have multiplied. Globus is always multiplied on host plant, so we have used maize as the host plant for multiplying the the fungi Globus species, and we have used as inoculum. We have used spores, then the infected root fragments, and combined we have given to, the quantity was 5 grams, which was given to the saplings. Then the fungus which was considered was Pestaloteopsis disseminata, because this is the fungus which is responsible, or which was responsible for the maximum leaf loss. So we have standardized what quantity of this is to be applied to the pot plant for challenging the plant or for initiating the disease, and then incorporation of the bioinoculants for controlling this disease. Now you see, this is these are the different treatments. Actually, in Assam, there are 8 varieties. They call it morphotypes. They are not called as varieties. They are called as morphotypes. So there are 8 morphotypes of the Muga host plant, and then they were tested by, actually it is by mistake Bacillus familis, it is by Pseudomonas species. So you can see the difference between the growth of the control and the treated plant. So here again, what we did after we applied into the pot, we calculated the percent disease index, and what is very important here is all the morphotypes, they did not respond to the pathogen uniformly. Their response was different, and therefore, what from here, this morphotype S6, it was the most important morphotype which responded to the treatment, means biocontrol agent, and the disease incidence compared to the control was very, very low. This is total. We also calculated, means also estimated the total chlorophyll content of the bioformulation treated plants. So all the 8 morphotypes, they were tested, and as you have seen in case of percentage disease index, here also the morphotypes did not show uniform response. Their response was entirely different. This was total protein content because it was very, very important for the quality of the leaves. This is reducing sugar because it will again it will have impact on the quality of the insect, means growth of the insect and the quality of the silk. Then what we did in order to find out whether resistance has been induced or not, we did a lot of estimation of lot of defense enzymes. Now here I am showing two data. One is total phenol and peroxidase activity. So you can see that the different treatments, PGPR, PGPF, AMF separately, and the combined inoculation. So in each and every case, the impact of combined inoculation was better compared to when these organisms, they were inoculated individually. So now, why should we use biological control? So the chemical pesticides, you know, they have implicated. I had just shown you what we did. Now I am going to the general thing. Chemical pesticides, they have affected the ecology, they have affected the environment, they have affected the human health. They require yearly treatment because it is not that if you have, if the farmer, they have applied fertilizers, these pesticides, insecticides or other control chemicals this year, they won't have to give it for next year. No, every year they will have to give, and they are usually broad spectrum. It is not that they will only affect the target organism, they will equally affect. There are a lot of reports where it has been observed that they will equally affect the non-target organisms, and they can be toxic to both beneficial and pathogenic species. So therefore, what we need a biological control, which are non-toxic to human beings. They cannot contaminate water. They are host specific and only affect few species. Now, these are the environmental problems of pesticides, which have been reported by different workers all over the globe. And what is very important here is that biomagnification. Many pesticides, they undergo biomagnification. So it doesn't matter what quantity of the pesticide is being applied in the field, but finally the amount will increase as it moves through the food chain in the trophic level. Now, this is the market of pesticide. So you see the market is so huge. Now, this slide, what it shows the investment and return. So this is biological control. The small one is the biological control, and the large one is the pesticide application. So you see the cost and profit. So cost compared to cost in pesticide, profit is very high. In case of biological control, compared to cost, the profit is less. So when what are the problems? When we select commercially viable biocontrol agents, then the choice of pathosystem is very, very important. Means what I am pointing out here is the relationship between the pathogen and the organisms which need to be applied there. Then there are very few organisms which are tested for their capacity to control the diseases. You will find that in the case of microorganisms, most of the biological control is restricted to trichoderma. Trichoderma is widely used. Then whatever result we have got in case of microbial inoculants in the lab, that cannot be proportionately replicated in field conditions. This is one of the most important problems. And another important problem is the microbes, which if they are to be developed at the commercial level, then the selection criteria has not been considered, has not been taken into consideration. So to illustrate how and why to avoid these pitfalls, early stages of biocontrol product development. So these pitfalls, they are essential to be addressed before a proper commercially viable biocontrol strains are developed. So what are the challenges for the industry, biocontrol industry? There is not much expertise available. Market and products are very, very limited. And the involvement of science and technology, that also is restricted. And another important problem is regulation is not there. We do not know, nobody certifies that, okay, this is the product where biocontrol agents were used, or this is the product, what is the date of expiry, how long it can remain viable. All these things, they are not, there is no rules and regulation for that. So if we want to transfer this technology, so these three groups, these three steps are to be followed: the customers, the client group, and the customers. So this is the public domain, this is the government institution, which will finally go to the client group, and the patented, it will go to the customers. So there are different stages, like there is, in case of public domain, market analysis should be done, financing should be done, and then finally the product development will be there, which will go to the customers. Government, what should do? Government should find out the feasibility study, appropriation, product development, government should help, and then finally it will go to the customer. Then entrepreneur or company, they should do the market analysis, they should look for the financing, license for commercial product. I don't think, I have gone through the literature, I have not found any system of licensing so far as biological control agents are concerned. And then finally product development, and it will go to the customers. So what are the requirements for commercialization? Normally, if you look at the research, normally the research is not done on, means there is no institutional coordination as far as research is concerned. Individually, we are working everywhere. And there is financial support for R&D, but that is limited, and it is getting limited every other day. There is lack of practical technology. There is effective, which is effective against the pest. There is high benefit cost ratio, which I had shown you in the previous slide. And what is the safety for the environment if we are using, because even these days genetically modified organisms can also be used. So what are the safety concerns for the environment, that also is to be looked into. So these 6 aspects, they are very, very important for commercialization of biological control agents. Now, what are the shortcomings? The shortcomings are, I told you in the previous slide, the first shortcoming is all these biocontrol agents, they are selected on the basis of their results in the lab. And second aspect which is taken into consideration is how easy it is to mass cultivate them. Then the ecological parameters, actually there is no, or minimum emphasis is given on the ecological parameters. What are the ecological parameters? Like when they will be applied into the field, how long will they survive? What will be the intensity of their survival during the target period, and whether they will be preventing the persistence or not. Then another important problem is conflicting role of microbial inoculant traits. If you look at the traits, some of the traits, I will show you in the next slide, some of the traits are just conflicting from one another. And therefore, this also complicates the selection of microorganisms that functions or that will give you desired result in the field. And then there are a lot of reports where it has been shown that the actual result which is obtained in the lab cannot be replicated entirely into the field, and therefore the results are very unreliable. So these are the ongoing research challenges which all of us should take into account. So there is a need of feasibility of developing ideal soil microbial inoculant with current approaches. The latest scientific developments which are there that are to be incorporated before we really select an organism. Then there is a requirement of investment in this field by the agricultural biotech companies, and there is demand for effective inoculants with agriculturally relevant traits like nutrient mobilization, increase, the nutrient, basically when we had started working on the microbial inoculants, then basically what we see whether they are capable of mobilizing the nutrients or not. And then commercially useful microbes can be rapidly characterized and identified with new. We will have to use the omic tools for characterization and for characterization of commercially important microbes. Then this I have already told you, there is poor reliability. And now what is the future prospects? Most focus has been on free living rhizobacterial strains. Now what I have seen that no or less emphasis is given on other organisms. Like for free living rhizobacterial strains, if you look through the literature, then you will find that maximum work is on Pseudomonas and Bacillus. Whereas there is one group of organisms which remains inside the host, and they are called as endophytes. And these endophytes, they should be explored for their ability to reduce the diseases or they should be made a cardinal component for the management of plant diseases. So endophytes, because when they are residing in the host, they are not causing any disease. They are asymptomatically present, but there are a lot of theories about this. There are one school of thought which says that these endophytes, even they are responsible for the release of secondary metabolites which the plants are releasing. There are another school of thought which says that if they are not releasing the actual secondary metabolite, especially of medicinal importance or like antibiotics or other things, they are intervening into the biosynthetic pathway, and finally the final product is released by the plant. So when they are present there inside the host, asymptomatically, what are they doing? So there are a lot of reports where it has been shown that they are providing biotic and abiotic, they are alleviating the biotic and abiotic stresses which the host plant is coming across when they are present in the field. So another important thing is when they are inside the plant, they are already under a lot of selection pressure. So if you look at all these aspects, then this endophytic bacteria or endophytic fungi, they are one of the important candidates which should be incorporated for future microbial research for the development of productivity of crop, maybe as for mobilizing the nutrients or for improving the growth and yield of the plant, and at the same time for reducing the diseases. Now another important area is, so there should be techniques or there should be steps which should be taken for improving the efficacy of the biocontrol agent. Maybe genetic manipulation or whatever is required, that should be done. And then these microorganisms, they also produce antimicrobials. Like there are some nowadays, they are peptides. There are some bacteria which produce peptides, like Bacillus is used. So these peptides, they are antimicrobial in nature. So instead of applying the microbes, can we apply those peptides into the field so that we can control the disease? This is another area where a lot of work is going on. So identification of key antimicrobials produced by the superior agents. This is one antimicrobial diacetyl fluoroglucinol. This can be exploited for streamlining the strain recovery. Then Then the role of edaphic parameters, which are favorable for disease suppression, that should also be taken into account. Okay, these are the parameters which will help in controlling the disease. That should be taken into consideration. Then identifying different mechanisms of action to facilitate the combination stress. Because in my result, I have seen that when individual organisms they were used, so the result was not so good. It was good compared to control, but when combination of microbes were used, then the result you have already seen, that was significantly higher. So we can we incorporate bacteria, can we incorporate bacteria, fungi? So when we do this, whether they are acting synergistically or they are acting in a different way, maybe they can be antagonistic, but they can be they can be independent also. They will grow independently. So before we do this, this is to be this mechanistic part of the activity of these organisms. They should be worked out, and then there should be incorporation, application of biotechnology for further improvement of the strains. Then endophytic organisms, I have already told. Now this is one report which was published in the late 2019. Now here the life stages of microbial inoculants. So normally what happens, there is production, there is establishment, there is function, and there is downstream impact, like which we visualize in the form of growth of the plant. But the concept which is proposed by Kamniski et al. It was published in Trends in Biotechnology. Now here what he said, that these steps, they should be splitted to incorporate different traits together, so that the downstreaming impact will be better than what was in the top one. Now, depending upon this result, Kamniski et al., what he concluded that there cannot be tailor-made microbes which can be produced depending upon the requirement of individual crop or individual area. So to counter this, another article was published in Trends in Biotechnology. It was published by Avasthi in 2019, same year. So here, what he has said that by incorporating these, like one health card, then prediction means probability of drought, flood, heat shock, disease outburst, all these, then farmers' problem. When we incorporate all these, then there is possibility of tailoring microbes for a specific application, maybe specific tailored for specific to the plant need or the need of the area. So this is all I have to say. Thank you very much for giving me this opportunity. So I am through.
So thank you very much, Professor D.K. Jha, for your nice presentation. You have shown practically the management of foliar diseases of some plants using microbial consortium consisting of PGPR, PGPF, and AM fungi. Thank you, sir, for sharing your research experience and also highlighting some challenges and future prospects of use of microbes in plant disease management. So thank you so much.
Thank you.
Now, Professor L.C. Bora, Head Department of Plant Pathology, will deliver his keynote lecture. May I invite you to take your time, sir?
Yes.
Yes, we are making ready. Also, you are also doing, sir.
No. Is it coming?
Ah, yes, it's coming.
Is it coming?
Yes,
Is
yes.
it coming?
Yes, sir. It is visible.
Open your slides, sir.
It quotas. Just
Your time in another. Sir.
no better. No, it is again showing problem. You please okay. Okay, good afternoon now. From good morning to good afternoon. They are sorry for all the hiccups on the technology. And now I'm ready. Thank you, Dr. Jha, for giving some information that actually I am going to give now regarding organic agriculture and the developments that we have made in Assam Agricultural University and the type of research that needs to be done in the future. So the next, please. Next slide, please. Yes. So this is just how actually organic agriculture should be done, and the green approach for pest management, the primarily generally physical, mechanical, biological controls. These are the method used, and some light has been already shown by Dr. Jha. And biological control nowadays it is becoming very popular because of the effect of the chemicals on the environment. And in an area like this Northeast, it is very easy actually to explore these bioagents because it is abundantly found in the natural environment, soil, phytosphere, water. Then the suppressiveness of the soil act as a major source of identification. Wherever there is a natural type of control of plant diseases, generally, we can try to isolate the bioagents available in that particular locality. That is how actually we have done. Next, please. Okay. We'll have to look into this particular thought. A plant growing in a field is not an individual. It is a complex community with subtle and relatively constant partnership relationships. A well-structured and regulated community of microorganisms always escorted with the plant. And in addition to natural root colonizer, plant also recruit desired microorganisms in the rhizosphere when challenged by the pathogens. So beneficial ones are generally used in agriculture, and these beneficials generally are present as rhizospheric microorganisms and endophytic microorganisms. Next, please. Something has been already said regarding these endophytes by Dr. Jha now. He has made my work somewhat easier, Dr. Jha. Next one. Next slide, please. This is just a microplant crosstalk. And this interaction and nutritional security, how actually the microbes which are beneficial helped the plant increase root length, shoot length, biomass, yield, then resistance to phytopathogens and antioxidants. Then also at the same time, they access some of the nutrients, not exactly enhances actually. Some of the micronutrients or macronutrients which are not available are generally converted by the microorganisms and make it available to the plants. For example, the nodule bacteria we know very well, and plant growth promoting bacteria or plant growth promoting fungi as a whole, the plant growth promoting microbes. They help in phosphate solubilization, they help in indoleacetic acid production, they produce a siderophores by which actually the plant makes the nutrient like say iron unavailable for the pathogen. And some of the microbes which take role are vascular fungi, that is a fungi, then root nodule, plant growth promoting bacteria, and plant growth promoting fungi. Next, please. Yes, endophytes, I will not go into very detail about the endophytes, how actually their presence in the plant, maybe as a saprophyte or just as a neutral microorganisms without anything, they are present. Now, this is a new area where actually a lot of research at present has been initiated. And it has been proposed that they helps in disease management, production of plant growth regulators, then biological nitrogen fixation, induction of plant radiation, ISR, then drought tolerance, that is a very important aspect, then phosphate solubilization. So that means they are present inside the root plant system, maybe in the shoot system or maybe in the root system, or maybe they are present in the leaves. So they can take a lot of role in the development of the plant growth and suppression of the detrimental microorganisms. Next, please. Next slide. Yes. Now here just I am showing how actually the microorganisms are benefited as well as the plant is benefited. Whenever he is my friend, okay, then definitely the microbes generally provide certain source of nutrients, energy source. Similarly, the microbes also in return get the nutrient benefit as well as energy benefit. Next, please. Next slide, please.
Any issue?
It was second slide option. Now, endophytes, I'll continue, okay? I'll continue. Now, these endophytes, they may be fungi, they may be bacteria. And the bacteria generally are Bacillus enterobacter, Pseudomonas fluorescens, Bacillus brevis, then of course, Azospirillum species. Similarly, there are certain fungi like say Trichoderma is the major player. Then we have got some other very important fungi like say Aspergillus, Penicillium. They also play a very important role as endophyte. Now this is where actually this is the introduction part where actually the microbes has a very important role to play in development, plant health development, as well as the plant how actually they get benefits from the microbes, the beneficial bacteria as well as beneficial fungi. Now I will shift to the developments of Assam Agricultural University, where I have initiated some work along with my students for last about 22 years. I have also executed many projects funded by ICAR. I have executed some projects developed funded by ESP, DBT, UGC, like that. Even some Assam Agricultural University also, state also has funded in many projects. And out of that, some findings I want to just show. Now, as probably Dr. Banik has said in his introductory remark about me, that we have developed about 10 biopesticides. Yes, we have 10 biopesticide formulations we have developed, and 2 actinomycetes-based formulations are in pipeline. That means the actinomycetes we have already identified. Then the formulation technology we are just standardizing, and we have also tested their efficacy in the field. Now the fungi and the associated with the biopesticide formulations are the Trichodama harzianum, Trichodama viridi, Trichodama pararesi, Anisopoli, Beauveria bassiana, Verticillium licani, or Licanicillium licani nowadays it is called, then Pacillomyces fumosorens, then Actinomycetes just now I said. Then of course, the bacteria are mostly used, we are using Pseudomonas fluorescens, Bacillus subtilis, as well as Bacillus thuringiensis. Then at the same time, the entomology part also we are dealing with, that is the parasitoids, Trichogramma-like species we are using for management of insect pest. So if it is available, then please go ahead. Next 2 slides. These are photographs generally. If it is working. Okay. Now the technology or the bioformulations that we have developed, we have also named them like BioVir for Trichodama viridi, BioZium for Trichodama harzianum, BioMeta for Menisopoli, Matargia menisopoli, BioSona for Beauveria bassiana, BioLium for Verticillium licani, Bio4 PF that is becoming very, very popular in Assam, particularly because of its efficacy in management of wilt disease, bacterial wilt disease in solanaceous crops like vegetables, solanaceous crops like say, chili, tomato, brinjal, as well as Bhut Jolokia. Nowadays it is very, very popular among the farmers of Assam. Then Biozin PTB, that is effective against the rhizome rot of ginger. Then there is a consortia formulation of Biotime, where both insect as well as diseases can be managed by incorporation of Matargia menisopoli, which is a bioinsecticide, then Pseudomonas fluorescens and Trichodama harzianum, 3 different types of microorganisms. Then finally, we have got this BioGreen we have lately developed, which contains almost all different types of microorganisms. The students, particularly MSc as well as PhD students, they have worked on the compatibility studies, how bassiana is compatible or with anisopoli or not. It is compatible with Pseudomonas fluorescens or not, or it is compatible with phosphate solubilizing bacteria or not. Ultimately, these are the 10 different biopesticides, and of course, we have tested in the field, farmers field, as well as in our research station, as well as in KVK. Then we have targeted BioMeta, for example, that is anisopoli. We have targeted against red ants and termites, and in sugarcane as well as coconut, and we have got a significantly good result. Similarly, in Biosona, that is a Beauveria bassiana formulation isolated from Majuli of Assam, and this is effective against rice hispa, then leaf miner of citrus, then stem borer of sugarcane. Then Biolium, that is Licanicillium licani, that is effective against diseases of vegetables as well as nematode pest, very effective. Then Biovir, that is Trichoderma viridi, it is effective against bacterial and fungal wilt of vegetables. In banana, that is Fusarium rot, that is also effectively controlled. Anthracnose and stem rot of green gram we have tested. Then Biozium, bacterial and fungal wilt of vegetables and banana. Then Biotime, bacterial and fungal wilt of potato, vegetables, anthracnose, and stem rot of green gram, rhizome rot of ginger. So many diseases with chain pesticide, insect as well as pesticide, we could control, and that were tested in research station as well as in some pockets of farmers, some fields of farmers. Then next part is that we have started a pesticide production unit with the fund of DBT again. Then we have got a potential production capacity of 1 ton per month. But it is not sufficient, as you know, if the farmers require in huge quantity. So what we did next is that we have started developing some entrepreneurs by training them with the technology of biopesticide. And as a result of that, we have got in Assam about 5 different startups. They are producing, they have taken our technology, they are producing and they are distributing their products to the farmers. So now at least 30 tons per month we can easily produce with these entrepreneurs. Here we could produce only 1 ton per month. Then we started training. Now tea is a very important crop in Assam, everybody knows. So we started training the small tea growers in the field, as well as we invite the farmers to our station, that is our biopesticide center. So we train them to use the products so that they can go for production of organic tea. And as you know, organic tea is procuring more money for them, 10 times more than the general black tea. So organic green tea they are producing and they are getting good money by using our biopesticides. Of course, they are also using some traditional method, traditional herbs for controlling different management, different pest management. Then we have also started training the farmers, vegetable farmers, then rice farmers, the commercial crop producers, the ginger, then turmeric, bhut jolokia. These are we regard as commercial crop because of the money the farmers are getting from harvest of these particular crops. As you know, for commercialization, very important thing is registration by CIB. And we have already obtained registration of 2 biopesticides. One is Trichoderma viridi based Biovir and Trichoderma viridi plus some growth promoting microbe, that is BioGreen 5. These 2 we have already commercialized and we registered and are registered by CIB. And what we did next is we started giving this to big companies. Now 2 big company has been assigned to develop these biopesticides for us. And one is Orgamen Jorhat. Now they're producing about 100 tons now, 100 tons of biopesticide per month. Similarly, there is another organization in Shillong, SLRD. They are also producing this product and they are developing the organic growers of Meghalaya. Now, after that, of course, we are also not stopping. We are just working on certain areas like bioremediation, biodegradation. So bioremediation is a very important process, particularly in Assam, in one bank that is from South Bank, there is a lot of contamination of rice field by arsenic. And as a result of that, there are a lot of arsenic poisoning. So what we did actually, this is a research part, of course, we started isolating certain arsenic degrading bacteria. We have already isolated 2 different bacteria, both of a bacillus group, and that is under, of course, study. The study is going on. Some PhD workers are working on that. And as I have said, the rhizobacteria which are present has been already identified. Then another area we are working on is that consortia of methanotropic bacteria and plant growth promoting bacteria. That is rice field. There are a lot of, everybody know, in global warming, a lot of contribution is made through the rice cultivation. So if you can incorporate with our biopesticide or bioformulation certain this methanotropic bacteria, then definitely the reduction of methane emission will be reduced. So that is the idea based on which actually we are working. And one PhD student has already completing his research. He has also identified some methane eating bacteria he has identified. And he is developing, his other students are trying to develop the consortial formulation of methane eating bacteria. Then expand products. Plant products, that is Jatropha. It is a very, it has got certain properties of inhibition of the pathogens. So we use Jatropha, and along with that, we have combined some microbial antagonists, like we have tested Beauveria bassiana. So this Beauveria bassiana, it is somewhat compatible with Jatropha oil. We have tested 0.1%, then 1%, we have tested 2%, even at 3%, so they are compatible. So this Jatropha oil and microbial antagonists combined can be applied for management of pest, particularly insect pest. So this is some of the research. Then we have developed many books, biopesticide information brochures, booklets, handbooks for the utility of the farmers, guidebook. Then also we have sold through our biopesticide production unit about 29.35 metric tons and 600 liters, and as a result of which we have earned some money for the university, about 13 lakhs of money in last 2 or 3 years. Then we have applied patents. This is a mandatory for any technology development. We have applied for 3 patents. Then the approval we have already obtained from National Biodiversity Authority, deposited the cultures to the MTCC. And new area of research, that is what actually I am going to say and conclude, is that micronutrients has got a very important role to play in the efficacy of the micronutrients in suppression of the disease. This is where actually we have proved in rice, where the BLB, bacterial leaf blight of rice, is managed by Pseudomonas fluorescens. And after application of Pseudomonas fluorescens, we have checked for the upregulation of genes. Then we have seen then gene XA1 and XA6 were upregulated. Similarly, one area where actually the hydroponics, whether these microorganisms will be effective in water condition. So this we have tested in lettuce. Then 2 diseases, Fusarium species and Ralstonia solanaceum, they could be controlled very effectively with the application of Trichoderma viridi based formulations. best, then followed by Ralstonia solenosa. That means in hydroponic condition also, this bioformulation will be very effective. Yes, consortial formulation I said about, this is the effect of the BioGreen 5 in tea for production of organic tea. Very, very effective. These are the two bacteria we have already identified, Penibacillus species and Bacillus species, which were actually very effective in reduction of the arsenic. Then Erod germplasm of Northeast is a very, very important fact, and the assessment of host resistance by application of bioformulation we have tested. There are as many as near to about 100 different types of germplasms of eroids present in some surrounding states, and most are affected by Phytophthora as well as Xanthomonas. These are the two blight-causing bacteria and blight-causing fungus. And our antagonists so very, very good effective against these two pathogens. Yes, this is the bioprospecting of actinobacteria. Why actually we require this actinobacteria is that in deep water rice condition and in saline rice conditions, generally water is always there. So this actinobacteria generally plays a good role in that particular condition, and particularly because we have isolated many, but out of that we are showing 5 which are producing very good antibiotics, and we have already identified those antibiotics produced by the actinobacteria, and we are working on the genes responsible for this production of these antibiotics, genes responsible for the enzymes which are actually produced by actinobacteria and resulting into production of antibiotics. Then another area where actually we need to think about biopesticide is that they should be risk-free to the environment. Like in case of chemicals, we are saying, yes, they are not environment friendly. But in case of biopesticide also, there may be certain area where actually they may be harmful biopesticide, because some insecticide that is entomopathogens generally we use for production of biopesticide. So certain very, very beneficial insects which are present in the nature, they should not be harmed by this biopesticide. So that is one area where we are working with the title Risk Assessment of Non-Target Effect of Microbial Bioformulation, development of risk-free biopesticide and healthy food. Yes, endophytes, we have initiated work on endophytes. Then, of course, that is all about. I would like to share with you. Sorry, many disturbances were made, and you could tolerate. Thank you for your patience. Thank you very much.
Thank you very much, sir, Professor L.C. Bora, for your beautiful and attractive presentation on your achievements with microbial biopesticides and their commercialization. You have also talked about bioremediation, use of microbes for reducing the problem of methane in the environment. You talked about plant products. You developed products, bioformulations for tea gardens, and also you are working on bioprospecting. So we are very glad to have you as a keynote lecturer in this symposium, virtual symposium of IPS Northeast Zone. So thank you very much, sir, for this beautiful presentation.
Thank you.
Thank you, Dr. Bonik. Can I have a word
Yes, sir.
for both the speakers? And I don't know whether you have that in agenda or not. Can I take a few minutes, 1 or 2 minutes?
It's okay, sir. Please.
I must congratulate Dr. D.K. Jha as well as Dr. L.C. Bora. I read Dr. Bora's paper yesterday, and for both of you, partly I missed Dr. Jha's presentation because I had to deal with our experts who have come for ASRB. And but then mostly I attended the entire part of both of you have given very, very informative work, and I was not really aware of so much work. And now I feel that future of organic cultivation is very safe in your hands. And I can see Dr. Vora's presentation. Dr. Jha, congratulations for a very nice overview of the presentation, which which was also there with Dr. Vora's presentation. Dr. Bora, you very rightly suggested some of the things, because when I was there in this in ICR, few things, as you said something about nowadays plant protection does not mean only pathogens thing. We also have to work for insect pests under crop protected cultivation. Like
Right. Right.
you have touched a part of it, but when you go abroad, if you see some companies like Copper in New Zealand and all those things, they are still um, requesting, and we are also giving permits to import some of the biocontrol agents, like you said about predators, parasitoids, and certain predatory mites, which are very important under this protected cultivation, like Phytocillus amblicius and Anisius, such kind of this predatory mites and other insect parasitoids are very important. Some of you also may take up those things, because otherwise we are importing. There are technologies they have, but we do not have for mass manufacturing. Even SBA does not have. If they have, but it is not growing. So protected cultivation is under a big limitation. You said about many of them. I'm very glad to see that you have registered them. One thing, let me tell you, that the biopesticide, only when they are registered, then only you can give in your package of practice. So
Right.
whenever we are applying, kindly do that. Your IPR should be there, and you must submit, and then you should have you must have developed a fingerprint, so that you can retain and you can find, and you can keep a track of your well. Now the days are coming when lots of activities will be done on this, and we have recently developed guidelines for biostimulants. When you do something, biostimulants are those, they are also microbial, maybe microbial, they may be certain natural product, they may be certain botanicals, which are improving the physiology of plant and thereby making resistant. But moment we say that in the biostimulants we will be adding biopesticides and your PGPR, then it will be governed by CIBRC rule when
Right.
we combine these two. So there are separate guidelines which have been made by government. I was lucky to be a chair on that committee, and I developed some guidelines because people were making huge 600 crores worth of fictitious and spurious biostimulants, or they are mixing some certain fungicides, and they are mixing some biopesticides. So those things needs to be covered, and there are separate guidelines. You have to again go through. Then I also try to approach ICMR for registration process. If they can, if one trichoderma is given, why we need to do all trichoderma, say trichoderma viridi or say argenum or other trichoderma resi have come recently. One institute has developed a path-breaking consortium with trichoderma resi, for which stakeholders are coming from abroad, from India, to require those to allow those things. But then, can we develop certain clinical determinants, so that all the biopesticide registration need not go with all the six-pack studies which are presently required by CIBRC? So if we can determine that, like in case of Bacillomyces liliaceus, we have so far, which is called now Purpureocilium liliaceus, which is a nematicidal, and which is very important for Northeast region. One, I would like that the BioV, or which you have made, if
IOB.
it can be tested under AICRP biological control, to see what is the specificity. Is it very good, although it is good for your region, That was what I in my introductory remark I said, that it's not that one thing which is found good in tropical region will also be found good. Now you have one which is good for your region. So you can also see whether and now the government of India has made a rule by which you can give it to somebody else who in turn can become also 9. Now you are 9(3) resistant on that. You are a original resistant.
Right.
But for promotion of it, Government of India only in case of biopesticide have made something very detrimental, which all of you should come forward to say that what they have said, that moment you give to another company, he has and you share your toxicological properties and all what you have used for those registrations, he will be the owner of that, and he can also have the same power as a original resistant. So what he will do, what a person means, now the biopesticide which you tested, maybe it is good for your region. This will proliferate spurious one, somebody from Kashmir or somebody from maybe Tamil Nadu in tropical conditions. He will show that he has taken by paying you 2 lakh rupees as a cost of the technology, and he might be using his own technology there. He might be using his own trichoderma, because government does not have a paraphernalia to check the fingerprint for all of them. But to save his all he will not develop any registration. He will not. He will say, I am using Dr. Bora's strain, because he has bought and you have already given. But
Right.
under the garb of your strain, he will avoid doing all those studies which you have done, maybe 12 lakh worth of studies, which is required for registration. And he will just, for name's sake, he will show that it is being used. Your product is being used. So in doing so, it will proliferate spurious kind of thing. So you need to also check through All India Coordinated Research Project on Biological Control whether your product is good for all over the country or only it is good for your region where you have recommended. So then you say to your prospective people, otherwise farmers will lose faith. When your strain is used maybe in tropical region, they will say, no, it does not work, while it is working very good in your hand. So one thing is this one we have to be particular. Second, I would like to say that you please try to develop a determinant of clinical determinant, which is required for it. Not all acute inhalation, acute toxicity, acute, so all those things. So if it is if it is Bacillomyces lilaricinus or Purpureocilium lilaricinus, which is now called, so I have fixed the registration requirement as bacillotoxin production. Whether that strain has a bacillotoxin production ability, only then it has a clinical concern. So all other strains which are bacillotoxin minus, we don't have any problem. Second, another issue which I know, because you are very eminent in this and you have gone a deep way, your students. Now, Codex Committee on Pesticide Registration, they have made a EWG, Electronic Working Group, where till last year I was co-chair to Libya in that country. And we have been given the responsibility to see the use of biopesticides for the products, for the crops, for protection of the crop, where MRL is not a concern. See, biopesticide, MRL is not a concern, like chemical pesticide. Maximum residual limit is not required. However, Grape Research Institute has developed a biopesticide or a microbe which they spray terminally at the time of terminal harvest, so which eats away all the pesticides on the surface. But then, on one hand, it is good that it is degrading pesticide, a residue toxicity of pesticide. But on the other hand, Codex is trying to develop some kind of limitation or some kind of guideline to see whether if there are millions of molecules covering of this biopesticide covering on the surface, what clinical issue it might raise if somebody consumes that, because when it is terminally used and it consumes. So what is the allowability of the CFU which should be there? Whether they degrade. So such kind of study also you can see sometimes, because in nature when bio control is happening, it may be due to the propagules might be present 10 to 3, 10 to 4, 10 to 5. But while when we are using, we might be using up to 10 to 9 CFU per ml. So whether that much is not should not be interfering with the human consumption. So those kind of issues also will come. So similarly, in case of nematicide, because we are banning most of the nematicide, carbofuran has been banned. Now it is slated, it is going to be banned. It is slated for phase out, means by draft pesticide committee. And we have developed this Pacillobacillus, we have 2 new nematicide, but Poconia or Pochonia and Pasturella, these are other which are very important, because we don't have any nematicide. Kindly work upon it also, because now under changing this cropping patterns, nematode has become a very big issue. Another issue which very good work which you have said is arsenic, means degrading bacteria. With great difficulty, India to let our, you will be, some of you will be knowing, but some of you may not be knowing, that we are exporting basmati rice worth 20,000 crores to every year. But equal amount of other rice is also being exported. Nobody talks about it. We in Delhi, because it is grown only in North India and is a prized possession product, so we say only about basmati export, but we are not telling about the other rice-based export. So there, arsenic is and in northeastern and eastern region, arsenic is one of the issues. So last time in Codex Committee on Contaminants in Food, arsenic was an issue. The FAO and WHO was trying to fix the value as 200 ppb. With our involvement, we said no. Under natural and geogenic conditions, arsenic is there at a higher concentration also, and which is a natural process. So we cannot keep 200 ppb. So it has been raised to 350 ppb. But FAO and WHO has said that then you try to develop an SOP by which this residue of arsenic, which is found to the level of about 600 ppb also in India, it should be reduced not only to 350 ppb, but much below that. So your bacteria may be of great importance, and you kindly see and publish it and prove it in a multi location test that it does the job under field conditions. Then we can tell the countries like Japan, FAO, WHO that we have developed an SOP where the world can use that technology for that, and you can also give this technology. So these are few things, small things, because this is a great opportunity to apprise many of our colleagues and who are having students, so to see on these aspects. Thank you. Both of you have done so wonderfully well, and I am very hopeful now biostimulants will, or the biopesticides will not only be a lip service. It will go. Till now, we have only not crossed more than 5% area under use of biointensive IPM. We want, even Planning Commission wanted that India should go 100% biointensive IPM, but biointensive IPM is not going. So with the developments like what you have shown, we know the future is bright, and this particular sector is growing at a more rate than pesticide. It is growing at the rate of 12% compared to pesticide, which is growing at the rate of 8%. So biostimulants and biopesticides are having very potential future. Thank you all. Thank you so much.
Thank you. Thank you so much. Your suggestion has been noted. Thank you so much.
Thank you so much. Thank you.
Thank you, sir. Professor Dr. P.K. Chakravarti, President IPS, for your valuable input and setting the future priorities of research in this region. So thank you very much. And now we come to the closure of this technical session, keynote lecture session. We'll break for lunch and we'll come back at 1:30 for technical session 1.